got his undergrad diploma in biology and his PhD in biophysics from the University of Boulder, Switzerland. He then had spent a postdoctoral position at Scripps Research Institute in La Jolla and also in Europe in Dr. Heinz Gross' lab in Zurich.
Zurich, yeah.
Then he became a group leader in the Electron microscopy facility of the Macromolecular biology lab in Germany, in Heidelberg, Germany. And since 2006, he joined CU at MCDB, and he is the director of the 3D fine structure lab. Today, he will give us an overview of electron microscopies and how the images are formed in those microscopes. So,
yeah. Okay. All right. Thanks for coming. Yeah, my name is Andy Hoenger, and I'm over at MCDB. Actually, I'm in the Porter Science building. I don't know if you know, it makes an L shape. And then this very back, very down to the ground kind of area over there. So I'm going to talk to you about electron microscopy. I'll give you a bit of an introduction about image formation and 3D, a little bit of 3D image reconstruction. I hope that's all going to fit in 1 hour. It's a little bit a lot. So I tried to shorten it to the essential. Now, if you want to look at things, it's usually getting bloody expensive if they're either very big or extremely small. I don't know whether you have seen this guy that just was supposed to go in service about a couple of weeks ago, but actually they had to switch it off again. I think some cooling elements burned through already or something. Anyway, so we're not going to be in that league, and we're also not going to be in that league. So we're something like about here. Well, we really don't do scanning electron microscope here in the facility, but some people do it at CU. We mostly here in cellular electron microscopy and molecular kind of electron. So that's about our domain here, which is intact cells, entire cell structures, molecular assemblies, large molecular assemblies like these microtubules, docked in these particular cases with molecular motors, and do a little bit of atomic interpretation if that's possible of these things. So when you compare, I guess I'm not sure whether I'm preaching here a bit to the cryo, but when I compare now a light microscope with an electron microscope, obviously, you know, we have visible lens in the light glass lenses in the light microscope, visible light or UV light, sometimes ER, depends on what you do. In the electron microscope, we have magnetic lenses, so essentially the lens is a hole through a big chunk of copper, which is nothing more than a wire. Makes a huge magnetic field. The wavelengths in light is so roughly 300 to 800 nanometer. The wavelengths what we are dealing with in an electron microscope is about the 30th of an angstrom, depending a bit on what we call the accelerating voltage. At 100 kV, that's a routine microscope. So we accelerate our electrons with 100 kV, we get about 0.37 angstrom wavelengths. The source can be a tungsten, like in a normal light bulb. It can be a so-called lantern hexaborate crystal, or it can be a field emission cathode. What you're going to choose for is to some extent a little bit how much money you have available, and the other thing is the kind of coherence you want in your beam. A tungsten is more, let's say, comparable to a white light bulb what we have here, a couple of dollars per filament. LAB6 is a little bit better, about $1,000 or $2,000. And the field emission cathode comes close to a laser in coherence, as in really emitting a very distinct wavelength. But actually you pay about $500,000 easy for that kind of thing. The resolution limit we have in the electron microscope is about an angstrom these days. It's actually even a bit better, about 0.8 or so angstroms. The major, so you see when you look at the light, in this case here, the resolution limit is really these days the physical limits what we have in lenses. In the case of an electron microscope, we actually have comparably lousy lenses because we are far away from the diffraction limit of what the lens could theoretically do. So, okay, what is the advantage over the light microscope? Obviously, we have much more higher resolution. We also have a very long focal depth, so if you're familiar with confocal microscopy, where you focus through an object, you get a 3D thing, this we cannot do because our focal depth is too long. So we just get a whole virus or a whole whatever in focus from top to bottom. So we cannot. That has in fact to do with the relatively bad performance what these lenses are doing. But nevertheless, it allows us to study molecular detail, and then we have cryo techniques available, so we can actually freeze the distinct states. The drawback over a light microscope is we have a vacuum environment, so we can really only look at things which somehow sustain in the vacuum. There are very new experiments now with sort of liquid chambers which you can insert in the microscope. That's not supposed to be. Which you can insert in the microscope, but that's a brand new kind of way to go, which hasn't really resulted much in much detail yet. So the principal two types of electron microscopes, in a way there's a third one, which is sort of a hybrid between the two. So we have a scanning electron microscope and we have a transmission electron microscope. In our facility here, we mostly work with, or actually almost exclusively work with the transmission type. I think in engineering you also have a couple of SEMs available. Also in biology, in Ramley biology, they have an ACM if you need that kind of machine at some point. So you have an electron source, as I said already before. Can also be a field emission in the case of a scanner. Can be a tungsten, whatever. Typically the acceleration voltage is a little lower, it's only about 30 or so, 40 kilovolt. While in the case of a transmission, we typically go up to 300 kilovolts these days. We have a condenser, which makes us essentially the beam parallel. That's the main reason of a condenser. That's about the same. So roughly up to here, these microscopes are more or less the same. But then the key difference in a scanning electron microscope is you use a lens, not to make an image. So the lens is not an imaging lens in that sense, like you have it in another microscope. The only thing what the lens really is doing is it focuses the beam on a very tiny spot, and then you have deflection coils which scan the object up and down. And for each scanning increment, you measure a couple of events. For instance, secondary electrons or backscattered electrons or all kinds of stuff. So you don't really do an image with the lens itself, okay? This is different in the normal or the transmission. Let's not say the normal. So we have here our stage with the specimen. Okay, what I forgot to say, of course, is in this case the specimen can be large. Can be as large as a finger, or it can be really large. It can be a stone, or can be anything like that. It does not need to be transmissible. In the case of a transmission microscope, typically 300 to 400 nanometer maximum thickness. And if you get more than that, you get into problems with multiple scattering, and at some point it's just pitch dark and you don't see anything. And then we have an objective lens, or a real objective lens with apertures and everything, as you have it in a camera, essentially. And the projector lens as well, which then projects the image on a fluorescent screen, typically, which we can observe by eye directly. Or what we typically do these days, we have CCD cameras, so we have film. The older version is film. Sometimes we still use film. For some application, film are still actually very good. But typically we have these days CCD cameras up to 16 megapixels. And 64 megapixel is the newest one. But again, it's a price tag behind it, which is almost $1 million. So I guess for now we're going to go with our 16 megapixel cameras. So this is a typical scanner, EM image. So you see it's a big thing. So this is a mosquito in this case. Not transmissible, so you measure surface effects, so you measure the effect of the electron beam on the surface, all these kind of things, what happens there. Unfortunately for molecular resolution, typically the scanning EMs are not powerful enough. So they typically get to about 30 or 40 nanometers, oh, now let's say angstroms, 30 to 40 angstroms, but they don't really get to clear molecular resolution. And regular SEMs are maybe 10 nanometers, 20 nanometers in resolution. So that's good and right for these kind of things, but it's not enough for actually getting molecular resolution. These are just a few. I don't know whether you have ever seen an electron microscope, but just a few examples here. That's the cheapest kind you can buy at the moment. That's on a regular routine microscope, I would say, with pretty good quality. They're actually not exactly the newest anymore, in case you have looked at newer ones. But the principle is, of course, still the same. We have the gun up here. This is an FEG gun in this case. This is a regular gun. We have the stage, in this case here. That's where you do this little hole, you put the sample in, and what you observe on a little rod. In this case, the stage is only this little bit. And then we have here the aperture, the objective area, condenser area up here, objective area here, and the projector down here. And then in this chamber here, we can observe our images. And we either have cameras which come in here, which act more or less like a wide-angle camera, like that one too, or we have cameras at the bottom, which is then a more precision camera. You can imagine the beam spreads, so the field of view up here is much bigger than the field of view down there, but obviously the resolution is worse here than it is down here. So it depends a bit the trade-off what you want. If you want a high resolution, then you have the camera at the bottom. If you want to have a big overview, then you have the camera at this position here, the wide-angle port. Then what I said is an electron microscope can easily give you 2 angstroms, 1 angstrom resolution. These are gold grains, and these lattice lines here, this is the 2 angstrom gold lattice, what you see here. So we have we can actually in principle easily produce a 2 angstrom image. Now the only problem is this is okay for something like gold, but on a biological sample that's far from being possible. And the problem is this, that biological sample beam in an electron microscope, unlike a light beam, is obviously this kind of dualism. You know, we have electrons, which in some sense behave like a wave, so we can describe them as a wave. But they can also ionize our samples. I mean, they can so elastic. The kind of interaction we want is elastical interaction. That's what we look for. But that's not what always happens. We also have inelastic interaction, which means the electrons actually hit the sample, get stuck, or if you wish, and ionize the sample and destroy the sample. And for a gold sample like this is no big deal. But a biological sample is getting destroyed within seconds. So getting the resolution there is a tricky business. It's not that the microscope wouldn't do it, but it's just that the sample gets basically burned before we can collect enough signal to get the actual resolution we would like to have. So if you have a thick sample like in an SEM, then what's happening is sort of something like this. You have an incoming primary beam. That thing has a certain energy, like let's say 30 electron kV, roughly. That beam hits the surface, and these electrons at that speed sort of distribute in an area like this, perhaps, so let's say. In an area like this, well, actually all over the place, they generate what is called secondary electrons, which have a much lower energy, typically only about like this. All the electrons which are produced down here, they just don't really make it out to the orbit. So for the image information, they're actually irrelevant. So it's mostly these kind of things about that volume which is relevant for so-called secondary electrons. There may be, however, be secondary electrons also generated in this kind of volume here or here, but they're typically much less. So for an image, like I showed you before, what you collect is these kind of electrons from that volume. And you can actually, because they have a pretty low energy, you can sort of suck them off. I would say you just pull a magnet in there with a certain power, and the electrons would actually fly towards that kind of magnet. So you can collect them over there, picture point by picture point. If you want to, you can actually also collect backscattered electrons. They come from a much wider area, so the image is certainly not as good as from the so-called secondary electrons. So the backscattered electron, and then the other thing is you need an annual ring detector to actually get them, because they're too fast. You cannot really get them out of their trajectory. So you use an annual ring detector to actually collect these things. What you typically do with backscattered electrons is if you have a label on the surface, like a gold label, for instance, or something like this, because then on the gold labels you get a lot of backscattered signal, and you see that kind of gold label very well. But for a typical biological structure, the backscattered electron signal is usually not used. If the object is thin enough, you can either, when you scan, you can do the same thing also with transmitted electrons. So you can make a dark field detector, an annual dark field detector. You can again have a backscatter if you wish, which you typically don't have though. And if you actually have a flashlight, what you typically have is just an open beam. Then you just use a bright field, as you use film image plate CCDs, as what we call a bright field detector. what we then sometimes have is an additional so-called energy filter. And the energy filter, in principle, is a simple thing. It is a tube which is bent 90 degrees. And you know, when you come with a charged particle and you hit the magnetic field, you get a deflection. That's the same thing actually the lenses work too. So you hit an electric field and our magnetic field, and you get a deflection. And depending on your speed, you can just change that magnetic field here. Depending on your your speed, they either make it through, or they hit the walls. If they're too slow, they hit the wall up here, and if they're too fast, they hit the wall down here. And if they are coming just exactly right, then you get them through. And that's a good way of selecting elastically scattered electrons, the ones we actually want, from the inelastically scattered electrons, because they have actually lost energy, so they're not at the same speed anymore. And also secondary electrons. All the secondary electrons are typically not a big problem. When you do this energy filter, that's again this energy filter system, and then you can actually have your CCD back here, and you collect your image back there. And with this thing, you can do a few. Oops, I forget this. You can do a few interesting things. Depending on, you can, for instance, as I said just before, you can collect these elastically scattered electrons which has not exchanged any energy. So they're the so-called zero loss picture. So that's something like this here. Now here, what we have here, we have actually a mix between iron oxide and titanium oxide. And that here would be the zero loss image. But some electrons or some atoms break down the speed of electrons in a very characteristic way. And this you can use in this case for actually detecting areas which have iron or whatever, oxide or something like this. So see here, for instance, we have this kind of energy loss curve, and 500 something electron volt is a typical band for oxygen. 708 is a typical band for iron. When you are now in this so-called energy loss, zero loss kind of mode, you just have the slit here in the center, and the other ones sort of get deflected and hit an aperture here. But you can actually change that slit to a particular loss number, like here. And then you can, for instance, image iron. So you have here a signal for iron, which is separated from the rest. You can get a signal for titanium, and you can get a signal for oxide or oxygen. And this is what we call element mapping. So you can actually detect specific elements in an element like this. So that's particularly interesting for material scientists. That's in biology not so much used because you need a lot of those. So we have a couple of ways of preparing our sample. I told you already before, we have to go to vacuum, which means we have to make our sample stable in the vacuum. So a liquid drop would evaporate, cannot be. So one thing we do, which is the really simplest way and the easiest, basically everybody who comes on a train with us, you're going to tell them do negative staining first. So what you do is you soak your specimen, typically your biological specimen in this case. I'm talking now, just to make this clear, I'm a biologist, okay? So from now on, what I'm talking is mostly biological samples. Proteins, protein assembly as a macromolecular assembly, and these kind of things. We don't really deal, if you want to look at iron or gold, obviously you don't need the kind of staining because these things are solid already again. But the protein solution or protein aggregate is typically in solution, so you have to somehow dehydrate it, or you have to keep it frozen, which I just come to it in a second. So in this case, we would dehydrate it, which means we soak it with a heavy metal salt. And this is in this case uranyl acetate, for instance, or ammonium molybdate, are pretty popular salts. And what they actually do is it's a little bit as if you have a broken arm and you have a cast, and you actually image your cast, okay? Not the arm itself, but you image the cast around it. So that's this negative staining kind of thing. That's why it's called negative staining. So the protein is less electron dense than the stain around it. So what can you do with this? You can actually get 2D and with some tricks 3D information to about 1 to 2 nanometer resolution. 1 is optimistic, 2 is realistic. You don't really need much of a specific equipment. I mean, it's very fast and it produces a lot of contrast. It has a couple of drawbacks though. The problem is you soak, as I said, you soak with this heavy metal salt. That heavy metal salt is not necessarily a favorable solution for a protein. Some proteins don't like it. You have also usually not exactly the right pH, and you dry in the whole thing. So you may get drying artifacts. That's about how does the specimen look like. What we're looking at here is you're going to see a lot of microtubules. I'm sorry for that, because that's my field. So all microtubule-like kind of things. But this is a microtubule which is sort of flattened open. Also basically, if you take the tube and you unflatten the wall to a two-dimensional sheet. And that's a typical image in negative staining. So you see these gaps, so the protein is white. And these gaps are canals or whatever in between which are filled with stain. That's why they come up dark. Also here again, this is the protein here, these blobs. And these big things here is actually stain-filled holes. So that's why the whole thing is called negative stain. But as I said, it is easy. It gives you an idea about how a protein may look like relatively fast. And it has a lot of contrast. The other thing what we do now for best possible preservation is what we call embedding in vitro size. So essentially what we do is we take our physiological buffer, we take our samples in the physiological buffer, and that's just what we freeze right away. We don't really do much with it. Now obviously this has the advantage that you are in the correct buffer. You can modify the concentration quite easily. And then the other thing is also you have a quick freeze. You have a quick freeze effect. So you just freeze anything in a conformational state. Sometimes if you can coordinate that a typical conformational state, then you will be able to actually look at distinct conformational states in a protein. So the specimen remains fully hydrated, and the contrast is actually generated by the density difference from protein to water or solution, whatever you have, which is typically about a factor of 1.2 to 1.3. So it's not a lot. Like compared to negative staining, we have about a factor of 10. So we have not that much contrast there, but we do have enough usually to work with. But it can be a problem. So again, we get 2D and 3D information, but in this case, we actually can get it to atomic resolution. And the world record in an EM reconstruction of a protein today is about 1.9 angstroms. So it's pretty close to atomic, or yeah. Then again, the specimen preservation is a very physiological conditions, and typically fewer distortions because you don't dry in, so you don't have flattening, all that kind of stuff. And the quick freezing may allow you to capture dynamic states. That may or may not work. And of course, the biggest advantage is you don't really look at the cast, which may be wrong in places where the stain doesn't reach, and so you actually reproduce the real protein shape as it is. Problem, low contrast, and it requires a fairly expensive equipment sometimes. So this is just the way we actually freeze, or we have frozen in the past, which actually still works very well, if you ask me. All you have is a solution of liquid ethane, which is now cooled here by liquid nitrogen. And you have some plunge freezer, so you mount a grid on the tip of this forceps. And you blot this thing and you drop the whole thing in the liquid, and that's it. And nowadays, for $80,000, you can buy this machine who does exactly the same. Sort of. Well, no, it's a little bit better. It keeps your sample at the desired temperature, at the desired humidity. That's essentially the big advantage of that thing, which is called VitroBot. But otherwise, it does exactly the same. The principle is exactly the same. Then what you want is not this, okay? This is a nice hexagonal crystal, the way you see it in snow or whatever. Any kind of ice you basically encounter is hexagonal in most cases. What we want is this. That is amorphous, which means we have no crystallinity. We have a state like a liquid, a frozen, like glass is a bit like that, an amorphous liquid essentially, which is just immobilized. This here is cubic ice. This is very beautiful cubic ice where you see the spots. This is the kind of cubic ice you typically encounter in your microscope if the freezing went bad. And then you get these kind of sharp lines based on the water molecules, which should really disappear in the case of frozen, of vitrification, where the water molecules are in all kinds of random orientations. And this is the kind of state you want, vitrification. And that's about how a microtubule again looks like in ice, compared to the structure before. Now this is not an open-end microtubule anymore. This is really a compact tube. And these little, so you see here the protofilaments, and these little T's which come off are 40 kilodalton motor protein. That's a kinesin motor head domain, which is about a protein blob of about 40 kilodalton. Now, as I say, you need a bit of equipment. This looks not like much, but again, it's in the order of a mid-sized car, about $30,000-$40,000 to buy that kind of holder. It's essentially nothing more than a liquid nitrogen dewar here. And then we have a rod, and here we have a liquid nitrogen filling chamber where we load the grid and the tip, and this tip then pulls out and goes into the microscope. So that's it. And the tip up here is cooled by this liquid nitrogen back here. What you reach, you usually keep your samples then at about minus 170 or so degree for roughly 2 to 3 hours, or even longer sometimes. So these again are these kind of microtubules. You see here, this is the way you would see them in a light microscope, so you can detect a microtubule easily with fluorescence. So you can single molecule detection is no big deal these days anymore in a light microscope, but it does not give you a structure, right? It gives you a detection. It tells you here is a microtubule, but it doesn't show you how the microtubule really looks like, except maybe for the lengthy kind of shape. These are kind of microtubules like we encounter them in an in vitro approach. Typically in vivo, you have that type here, which is a 13 protofilament microtubule. Very straight protofilaments, basically no super twist. That's the typical, I would say 80 to 90% of your microtubules in your body are actually of that type. Now for helical reconstruction, these kind of things are not usable. They have a kind of a lattice problem, which you cannot use for helical reconstruction. That's why we actually always used these kind of 15s. And they're sort of super twisted. You see here, then you get this kind of MRI. So these protofilaments are not going exactly straight anymore. They actually do a bit of a twist. And that gives you this distinct MRI feature. So you can actually, that's the way you can actually identify them. You can go to a micrograph and look at them. Okay, this is the right one we want. Then another thing what we also sometimes use is surface shadowing. In this case again, we freeze first, the same way we do it for cryo. But then we take the grids and we go to a sublimation chamber and get rid of the ice, also if a solid gas phase evaporation. And once the whole thing is dried out this way, we're going to shadow it with typically platinum or high resolution approaches is typically tantalum tungsten alloy, which we use. What can you do? Again, you get 2D information, 3D surface information, I will call it this time, to approximately 1 to 2 nanometers. Depends a bit on the grain size you can generate. There are even some better results, roughly 0.7 or so nanometers or 7 angstroms is currently about the record of reliable resolution. Again, it has the advantage of a cryo-preparation, and it allows to visualize also freeze fracture sites in case you want to do that. That's more for the cell biology, not so much for the molecular people. But cell biology, what they try to do, they freeze a tissue, they break it open, and they shadow that kind of surface, and then what you have is a replica of that particular surface as a shadowed image. Drawback, freeze drying may sometimes collapse the structure. Again, it's not a terribly cheap kind of way to do it. So see here, just as a direct comparison, this is an empty microtubule in ice, also frozen hydrated, also the direct cryo preparation. And this is the same thing with shadowing. So first of all, you see that thing is flat. It shouldn't be in principle, it should be still a cylinder, but it's not a cylinder, it actually collapsed. But then what you see is the surface. You see pretty nice, you see the protofilaments going here, you see some of these things going out. And here you see, while in this case you really look at the surface, in this case you should think about looking at an object which is made out of glass, and you look through the whole thing. So you see both, you see the back and the top, and you see the entire density superimposed. Well, in this case you really look at the surface only. And this is the same thing decorated with these motors. So we have these teeth again, and these teeth here, they show up as little lines going in this direction. So you see they cover entirely the protofilament, so we don't see the protofilaments anymore. Now we have this overlaying lattice which comes from these molecular motors, like in this case here, if you wish. So we see these lines, so to say. And in this case, we see these underlying protofilaments as a dominant feature. So if you want to get 3D information, we have to do a little bit of an effort. First of all, it sounds very easy. You want to have a couple of views from different directions, you merge them together, and you have your 3D, right? Now, the problem, of course, is that when I go to the microscope with something I don't know that this is a house up here. So I have essentially no clue how this looks like. So I have no clue how these things fit together. This is the major trick. So essentially, all kinds of 3D reconstruction methods, what we use in EM, build on that kind of fact that we produce different projections from as many angles as possible, and we back project them into a 3D space. And getting these projections right, knowing that this is the side, and knowing that this is from the roof, if you wish, or that this is from front, this is the tricky part of getting the information right. And there's a lot of bullshit in literature, believe me, where things got screwed up. But that's the very fundamental way of how we do an image reconstruction. As I said before, we cannot use the fact of focusing through, because we have a way too long focal depth. So we get everything in focus, so it doesn't help if we try to focus through an object. Unless the object is really huge, but then it's not really good for electron microscopy anymore. So the point is this, that no matter what kind of lens you use, your eyes, photo lens, electron microscopy lens, what the lens is always doing, always screws a dimension, okay, in the projection direction. Now this fish here is already screwed because of this lens up here, provided I would have a real fish up there, which I don't obviously, but you know what I mean? I mean, we have already one projection in this direction. Now I'm going to show you another projection in this direction. And what I end up is this here, which is all the densities along a line superimposed into one spot. And then I can back project as long as I want. I'm not going to get my 3D back anymore, right? So I have to get different views. Otherwise, this is not going to go. So on an image like this, you probably will tell me, oh, this is very easy. We know exactly what's in front and what's in the back. Now that, I must say, is your training. That's not the image, okay? This is because you know to look at these things and you know exactly, this is somewhere in the back. These people are much smaller than here my wife is much bigger, so they must be in front of us. But even artists in the past didn't have this really under control. So it's about around this time when people started to actually get perspective in the images. And this Konrad Witz, this is the Christophorus carrying cheeses over there. I don't know if you know the story, but anyway, it's one of the few first images actually, in fact, where we had a perspective, at least as far as we know in history. And you see all medieval images, they sort of screw up history quite a bit. I mean, they have a little bit of a perspective, but the artists really didn't, either they didn't worry, it's possible, or whatever, but they usually didn't paint in proper perspective ways. Or then they painted like this, you know, and here the perspective looks sort of right, but obviously that's a misleading illustration, and the illustration is only possible in 2D, because you get fooled by the fact that this is actually a 2D image and not a 3D. The same thing is true for this. So it would be good if that would be true, but it's unfortunately not the case. So what we need is, we need our different views, as I said, and then we can back project in space. And now in 2D space, we will do this, of course, in 3D space, and we get our image back. And we have our fish back. So, why, having said that, why do we get in an SEM this 3D looking image? Anybody an idea? This is no, this is not made by a lens. Okay, this is not a lens-based image mode. That's the difference. Because it's the shadow. No, it's not the shadowing. The shadowing is again a different thing. The shadowing makes you this 4D. The shadowing makes you a shadow, which gives you a pseudo feeling of a 3D. But that's not the case here. Here the electron beam scans, and depending on where it hits, it actually gets different signals. And we measure each particular point by point signal. but we don't really chase that image through a lens. Well, it has been chosen through a lens here, but we don't do that when you record the image. So don't get confused with the also tunneling microscopy, for instance, it's giving you a 3D sort of image, but obviously there is no lens involved. But when lenses are involved, that's all we have. Now the second thing we have, the second problem, is, and that's a particular electron microscopy issue. We have noisy, well, sometimes in light microscopy too, but we have noisy images. So we need actually to do something about the noisy images, and the easiest thing, if you can, is what we call image averaging, which means we have the image overlaid with noise, so we have a signal component and a noise component. Now, the way I would imagine this is if you watch TV, I don't know, somewhere in the mountains, and you have a bad reception or something like this, you get that snowy, flurry stuff. That's the noise, and the noise is a random thing. It has nothing to do with the signal in principle. It just overlays the signal. And the signal you can consider as a constant function, at least in an image. And when you add these things up, it's about, in an ideal case, your signal-to-noise ratio increases by a factor of the square root of, let's say when you take 100, you get about the 10-fold increase in signal-to-noise ratio, ideally. This row here, we have here this phase. It's here just random overlaid with noise. And it's just a simulation, of course. And you see, when I take 4 of them, 9 or whatever, 16 and so on, I get a better and better and better signal-to-noise ratio. Now, obviously, if this woman would make different faces each time, or look in different directions, I will get a mess, okay? I mean, that's what is not. It seems very logical if you speak in terms of a face, but in terms of a molecule, you never know. Molecules can be flexible. Molecules can be in different positions, even if you think they are in the right position. They may not necessarily be. And you don't really have this directly under control sometimes. So you have to be always aware of the fact that you get these kind of artifacts in your reconstructions. So what can we do with EM? Typically what we do is we can either do a tilt series reconstruction from crystals, from 2D crystals. We usually don't use 3D crystals. We only use let's say not only, but in most cases we use a 2D crystal. We can do a single particle averages or icosahedral particles, provided these particles are big enough. Or we can do a 3D helical reconstruction of a helical 3D reconstruction, provided that we know the helicality, or we have a helical object in front of us, like an actin filament, for instance, or the proper microtubule for helical reconstruction, or things like that. So all of these things are actually what we call averaging-based methods. The one who is not averaging-based is a tomographic reconstruction, where we actually take an object and we just scan different angles through in the microscope, and we record them all of exactly the same kind of specimen. I'm going to come to that in a sec. First go to 2D crystalline assemblies. They hold the world record for resolution, which actually I need to correct it. It's actually by 1.9 angstrom by now. Allows 3D reconstruction, typically of very small also something like a 40 kilodalton to even 20 kilodalton proteins can be reconstructed, provided you get them in a 2D crystal, okay? Straightforward combination with electron diffraction data. That may not be as important for you right now, but I'm going to show you in a minute what it means. And of course, it's the best method for low contrast specimens. What the drawbacks is, of course, you need to collect tilt series. And you have a missing cone, what is called a missing cone of resolution. The problem is this: when I have my specimen on a flat grid in the microscope, I would ideally be able to tilt in 90 degrees. But I cannot do that. I can only go to 60 or so for technical reasons, because I will need to get a beam through somehow, right? And if I go to 90 degrees, I would hit the copper like this. So I cannot do this to 90 degrees. And then this sounds quite simple, but it actually can be a major drag, because you have to get 2D crystals. And if you don't get 2D crystal, then you just don't do it, and that's it. So this is a crystal in, actually that's a negative stain crystal, I must admit. But what I wanted to show you here is a bit this. So this is the way a crystal looks in an electron microscope. And I'm not sure whether you actually see the crystalline lattice here. I don't know, do you see this from the back? It goes a bit like this, so you get some lines going this way, and maybe a little bit this way if you look. So it looks relatively disappointing on the first glance, right? So that's our 2D crystal in an electron microscope. Now we go often to Fourier space for this. So we can do a few things. We can actually filter the crystal pretty thoroughly. Based on the crossovers of these diffraction spots, we can actually make a filter which just takes the diffraction spots and nothing else. And what this means is the diffraction spots are corresponding to regular order structure. That's why we have a diffraction spot. So anything which overlays or is represented in a diffraction spot corresponds to regular structure, and in a sense corresponds to our crystal. Anything which goes in between is not interpretable because it's basically noise. So we have to treat it as noise. And if I do a filter like this, then my crystal already looks quite a bit better. Okay. So I have not added anything in this case. I have actually only subtracted the noise from the image. And in a crystal, you can do this right away because the crystal, in a sense, does averaging for you in a way that it provides these spots. Because these spots, as I said, is technically a sort of an averaging procedure where, based on the reproducibility of your positions, the more you have, the better these spots are getting, are becoming. Yeah. Now I can also actually screw up the resolution if I want, and if I go lower and lower in, I don't know how familiar you are with diffractions these days, but the lowest resolution in the center, the highest resolution is out here. And when I shrink this whole thing, I basically screw up the resolution. Okay. When I take this, you see, now you still have the 3 holes. I don't know, it depends how much you used to look at this thing. It may look even a bit better, but there's quite a bit of detail lost already, like these walls and so on, things like that. It's definitely getting more dramatic here. Now I don't even see these. This, by the way, just to tell you what this is a porine crystal. That's a bacterial porine. And that's a trimer, which has 3 holes. 3 relatively open holes in the outer membrane of a bacteria. So it's not a very specific kind of channel. It's just an open water channel. Well, anyway, so that's what this molecule is. But here you see we lose this 3 hole. This is the trimer here. And we have that not anymore. It gets even worse here. It gets really bad if you only take the center. So there's no information in the center anymore. Anyway, that's a good thing with crystals, so we can play around with these kind of things. Now that was a negative stain crystal, and in this case we actually have, as I said, we have a lot of contrast. We can focus in a relatively close to focus way. What that means is the following. When I go to an ice crystal, I have to defocus a bit to generate contrast. And what you have not seen in the other diffraction pattern, but what you see here is now these empty rings, or these so-called, we call these tone rings. So we have information here, and then we lose information, and then we have information again, but we have a phase shift. We have 180 degrees, sorry.
You're calling these 2D crystals, but we're trying to give 3D information. So can you explain?
Yeah. I just come to that. Just a second. Oh, okay, good. A 2D crystal is more a floor of tiles, for instance, where you just have one layer of regular arrays, but not in the along the z-axis.
How are you going to get 3D information then?
I'm going to tell you, just a sec. The 3D has nothing to do with the 3D of a crystal packing, basically. The 3D has to do with the fact how the molecule looks like.
Oh, it's a 2D array.
It's a 2D array of molecules, gotcha. But each molecule is a phase. I'm going to just a sec. So the thing is, in this case, we need to correct for these kind of rings, and this is called the contrast transfer function. That's a thing that's a lens-based problem what we have. And what people sometimes do is they collect this diffraction pattern by just collecting the diffraction pattern in the microscope by switching off the lens. So then they don't get an image anymore, but they get a diffraction pattern, and that diffraction pattern is not overlaid by these rings. So that can be very helpful because, as I say, sometimes you lose information in between here. And you have to correct, which means if I have a phase 180 degree here, I have a phase 0 here, which should be 180 degree. And then here the phase is correct again. So that's the way the thing is modulated. And if I do not correct, my image looks like this. And if I do correct, the image looks like this. There's quite a bit of difference, right? And in this case, actually, you can see quite a bit of detail. Here this is essentially uninterpretable, but here we see some helices here. This is actually a connexon from a gap, also from let's go gap junctions. And this is sort of a hexamer, and you see these kind of helices. So these little dots are helices. Now they are coincidentally exactly perpendicular to the axis, so that's a lucky case in that sense, of course. If the helix would be tilted or so, you wouldn't see it that clearly, for sure. Then you would see a smear or something. But you see, that's in the 2D reconstruction so far. And now for 3D. So we have our 2D crystal here, and we get a diffraction pattern in a plane. So we have diffraction spots. Also we have discrete information in the x and y direction, but we have continuous information in the z. I'm not sure whether you understand now what I'm saying. But with a crystal, when you diffract the crystal, you get essentially a destructive interference between the lattice lines and constructive interference at the spots, and that's why you see the spots. And this has to do with the fact that you repeat the information. But if you don't repeat the information, you get a continuous diffraction pattern. You don't get a discrete diffraction pattern, you get a continuous one. That's sometimes about a 2-hour course by itself, I know. So that means essentially this. When I have a 2D crystal like this now, such a packing, then we have discrete information in x and y, according to the lattice lines here, but we have continuous information in z. And now we have to collect this continuous information. And the way we collect this continuous information is by tilting the crystal. And when I tilt the crystal, I fill up the space with this plane here. So the plane in Fourier space essentially does the same thing as the planes do here. And these different views in projection, you basically should look at this in principle, not at this. These different views in projection correspond to continuous amplitude phase information along each of these lattice lines. And that is your 3D information, ultimately. There, go. So you see, again, we have a discrete spot, but in Z, along this axis, we have continuous phase and amplitude information. And that is our 3D information, ultimately. And that, bringing back correctly, gives you the 3D data set. And this connexon again. And the reason why I sometimes choose this thing is because this shows you very nicely how bad this missing wedge, well, maybe I should have said that again. You see here we have this missing wedge which we cannot reach because you cannot tilt higher than say 60 or so degrees. And because we cannot reach that missing wedge, we lose it. And you see now what that means in the structure is this orientation is pretty badly resolved, while this orientation is pretty good resolved. So that's this missing wedge. It's like a chewing gum, you know. So you have in the Z direction you lose quite a bit of resolution. With 60 degrees you lose about a factor of 1.5. Also, if you have 3 in X and Y, you have about 4.5 in Z. And that's the effect of me. So the resolution is non-isotropic. Now that's different when you do single particle reconstructions. You can actually get isotropic resolution here now, because now we have particles in all kinds of orientations. So we should get all our projections fully sampled from 0 to 90 degrees or whatever. And of course we don't need a regular assembly, we don't need a symmetry, although it actually can be good if you have one. And typically you don't need any tilting in the beam. Drawback, the particles have to be large. Nothing, I haven't seen a decent 3D reconstruction. Yeah, okay. A decent 3D reconstruction with particles below something like 300 kilodalton. So you can really not take any large things. So that's a cryo-EM reconstruction, another reconstruction. That's a reconstruction here, and that's just a raw data picture where you pick these individual particles. Now the trick here is you have to find these orientations to each other, and that again is a fairly complicated. We call it. Have you ever have heard the term sinograms or radon transform? That's essentially what you do in this case. Every particle, when I compare this particle with this particle in projection, when I project in all directions around here, and I project this one in all directions around here, they're going to have what is called a common line. One of these projections is exactly the same. And that's the kind of projection you're going to find, and they will tell you about the spatial relationship between the two particles. Icosahedral viruses, they in principle do the same thing. The only good thing here is you have the symmetry of the virus, which is usually composed of a 6-fold axis. It's composed of a 5-fold axis, well, actually, I say this way: the 6-fold axis is not mandatory. You can have an icosahedral particle all made out of 5-fold axes. So a soccer ball, everybody in Europe, I wouldn't have to ask this, but here, everybody knows what a soccer ball looks like, okay? Yeah. So the black ones alone are sufficient to make an icosahedral particle. And the more white ones you put in, these are the hexagons, right? The more of the white ones you put in, the bigger your particle becomes, but it doesn't necessarily change the symmetry. So here's the 5-fold axis from below, but it typically has a perfect match with symmetry. And the 6-fold axis, you typically have a mismatch, so you cannot symmetrize the 6-fold axis. And apart from that, it's essentially an icosahedral, as is essentially a single particle approach. The good thing again with cryo is you can actually see these internal, you can have internal layers which you can reconstruct, which you cannot do with negative stain, for instance. They are icosahedral virus reconstruction with negative stain, but they typically only stain in the outer structure, but not the inner kind of layers as you have here. So you say, let me see. Then I would say, I go quickly to tomography. Is that okay? 5 minutes? Because then I leave the rest out. So the good thing, as I said before, the reason why tomography is becoming more and more important is tomography is not based on averaging. All these kind of reconstructions have an averaging component in there, which you may or may not have directly under control, actually. You always think you have it under control, but you may not. Now, this is not the case with tomography. And while this here is now is a typical helical average structure, this is the real life in an actual muscle. So here we have a helical reconstruction of an actin filament in red, tropomyosin in blue, and the myosin heads in yellow, and the essential and regulatory light chains here. Now, you don't see it that clearly here, obviously, but you see the real case. And the real case, obviously, is not as regular as this is. So that's always a bit of trade-off what you want to see. If you want to see the real case, you may want to trade symmetry against seeing the real structure. So these are thick filaments here, and these are thin filaments. That's more or less the corresponding to the thin filament, which is these parts here. And these bridges are the myosin heads in this particular case. So they also did some docking and averaging later on that thing. So again, we have this issue with the projection and the missing wedge. What I typically can collect is, because again we collect now tilt series in the microscope, but not of different crystals all over the place. We just take one existing real feature, whatever that is, a Golgi, whatever. And this thing we cannot tilt and full. So we get this kind of loss in information. You see here we lose the striation and stuff like that. We would like to go on, but we cannot. The case of tomography again. And so you see this here. This is a tomographic reconstruction of the same virus you have seen before. Now when I go to X and Y, this looks pretty good. So you see the capsids and everything. That's why we tend to show only this. But in reality, we should also show this. And you see that the Z-axis is substantially less resolved than the X and Y axis is. That's the thing which you have to live with in tomography. And here too, you see that kind of that's a spirillum kind of bacteria. That of course should be exactly tube-like, but you see we lose this upper and the lower structures here because of that kind of missing wedge effect. 2 more slides. This is now again just the tomographic reconstruction of microtubules. So you see we get quite a bit of, now of course there's no averaging involved here, unlike this one here, that's a helical reconstruction. So blue is the tubulin, and these are these kinesin heads again. Here we see the kinesin heads as these little blobs, and we can actually go now up and down. Also this is always the lumen here. They are cut a little bit at an angle, so you cut through the bottom at some point and then through the top at the other. But these are these kind of 3D representation of this kind of structure. And then we can continue. We can actually average this together once we know much better what it is, because now we have a 3D, so we actually have this bit better under control. And then we can make an average structure like this. And in this case, for instance, unlike with a helical reconstruction, we can see things which we otherwise would not see, like for instance these kind of bridges and stuff. or the seam. You see, this is this lattice seam. So this is this interruption in the microtubule which screws up our helical reconstruction with most microtubules. That's why we cannot. That's why we use this 15th and not anything else, because we have this interruption, and that's not visible on the first glance. So you have no chance to see where this is. But now we can see that, and now we can actually get these kind of things back. So okay, good. I'm done. Thanks.
We have time for a couple of questions.
When you're recording tomographic scattering, how do you get phase? You're recording on phase. How do you record it?
Well, the thing is with an image is you have the phase in your image automatically. So we do not have this problem like the X-ray crystallographer have, which only look at the diffraction pattern. Because we have both. We have the diffraction. We calculate the diffraction pattern from the image typically, if we need one. But that gives us automatically the phase in a principle in a separate file, but it gives us the phase. Yeah.
When you're doing your tomographic reconstructions, you're doing a series of tilts.
Yes.
And let's say you do 100 tilts along one axis.
Mm-hmm.
You have to also do 100 tilts along the other axis to get
Ideally, yes.
images?
Ideally, yes. In reality, it depends a bit when you have a cryo object. Tomography has actually been primarily developed with plastic section, cellular preparations, and they can take quite a bit of those. And there it works pretty well. So we do what is called dual-axis tomography. So you take one and then you rotate the grid and you take another one. And that's typically about 300 images.
Wait, so you do a series of tilts like this and then a series of
Tilts like this,
not
yeah.
all angles in between, not all
No. So what you have is a missing pyramid instead of a missing cone, which is not so dramatically different actually. The missing wedge, if you only do one axis, you have a missing wedge, okay? And that's what we still usually have with cryo things, because for cryo the beam is really devastating. So we get usually about 100 to 150 images, and then the cryo sample is gone. And there's no point in trying to do the other thing, because that would be so much worse. So that's why we typically don't do it. That's why cryo is more suitable for these kind of more molecular studies, where you try to get the tomogram first, but then get rid of the missing wedge by averaging elements which you then can pick, which you then have much better under control. Good. Any other questions? If not.
Well, so we thank Professor Hoenger again.